Hi everyone, my name is Martin. I'm coming to you from Cologne in Germany, where I've had my lab for about 7 years now. And I've really been looking forward to this talk, especially since our world's been changing so much right now and everyone's worried. I was looking forward to an hour of just thinking about science today. So that's good, and I hope that's true for all of you as well. Now, I'm in Cologne. I have actually had my lab here for a while. We're very interested in longevity and protein homeostasis in particular. And I wanted to just introduce myself, and also saying that I will be moving to Cambridge very soon. So I think that's kind of cool to be a neighbor to you guys very soon. And also relocate to the UK. In fact, I've been offered a job by Altos Labs, so I'll be moving my lab there. Also, I would like to encourage everyone to turn on their cameras if they want to. I'm happy to see people here, and also if you have any questions, please do not hesitate to interrupt me. I'm very happy about taking questions. I won't probably see them all the time in the chat, but I'll be happy to be interrupted if anyone has questions. All right, good. So I guess we can get started if everybody's here and if people have trickled in by now. And I'm going to talk about mutagenesis screens and what we can find with using these in simple model organisms about metabolism and longevity. All right. So in terms of introduction, what I find very interesting is just this very basic slide. It's a bit geeky, but I'm sure you can deal with this. So essentially, this is showing life expectancy over a very long period of time. This is basically since the 17th century. We have life expectancy data here. Basically of average human populations in different parts of the world. And so then ever since the Industrial Revolution, life expectancy has increased drastically and in a linear fashion for a very long time. So it's a very long ongoing experiment where a linear change has been maintained over quite a significant period of time. Of course, it's very different things that contribute to life span extension at different times. You know, the advent of modern medicine, improvements in society and healthcare, of course, chief among those. But the interesting thing is that life expectancy seems to still be rising. There are many discussions what natural limits might be, but this is just a very interesting situation that we are in, because we sort of are in a place now where our life expectancy is higher than that of most people that have come before us. Now that is nice, but it comes sort of at a cost, and that is potential frailty. So as we age, the risk of very diverse diseases increases. And this is true across an astounding diversity of diseases, whose major driver seems to be age. So the idea here is that, and you can see this here, for example, in both cancer and also in dementia here, these are biologically unrelated diseases, one might think, but the major driver seems to be aging. So one motivation for me and in our lab to address aging and to be working in geroscience is really to understand the root cause of these very diverse diseases, and to actually promote health over in people over the long term. An interesting facet here is, for example, just showing this diversity of diseases that are driven and by aging and become more likely because of age, is the fact that if we were to be able to completely get rid of cancer, if cancer were to be a completely curable disease, life expectancy would only increase by about 2 years. Just showing that, people who have cancer most likely have other diseases that are very likely to be lethal than just a very short time later. And this problem is called multimorbidity, which is, of course, a huge problem, as most of us probably know. All right. So going forward, just a sec here. So in my lab, what we're addressing this complicated question through a sort of a simple set of principles. And one of these principles is that we're focusing on protein homeostasis. Protein homeostasis is a key driver is a key component of aging and the key hallmark of aging. We actually study longevity, if you will, as a phenotype. This is survival demographics here up on the right, where you can see that lifespan extension is a phenotype, is a characteristic of a whole population, and in fact, we try to study that as a phenotype of these organisms that we study. Just in terms of the approach as geneticists, we use point mutagenesis screens to provide optimal resolution in these screens, and I will tell you a lot about that aspect. But basically, we try to approach the biological questions we're interested in through a forward genetics approach, by which we can have sort of an unbiased, open approach and find interesting mechanisms through this genetic approach here. And then finally, we focus on conserved mechanisms, meaning that we're not only interested in what we can find, for example, in simple cultured cells, but we're working with worms and also with mice. So that's sort of the general overview of what we're doing. And I first want to introduce our key workhorse. This is the nematode Caenorhabditis elegans. It's a microscopic worm that only has about it's only about 1 millimeter in size, and it actually here it lives on the bacterial lawn that is its food, and it's a fantastic model organism because its genetics are very well organized, very well understood. It's also transparent, and so we can do very important work with it genetically. These worms are hermaphrodites, in fact, so this improves the capacity for us to do genetics. And finally, we can do RNA interference and also CRISPR-Cas9 to modify the genome. So this is a fantastic model organism if you are interested in basic biological processes, because also the worm is sort of the simplest model organism that we use in labs when it comes to multicellular organisms. All right, so we approach this work by basically generating mutants through chemical mutagenesis. So we have, if there's a question we want to follow, we have the opportunity to use DNA mutagenesis. This is a very old method to just chemically modify the DNA, which then generates a large collection of different mutants, here indicated by these colors, that all carry multiple mutations. And of course, many of these mutations will not have any consequences. Many of these mutations will have deleterious consequences, but some might actually have beneficial consequences. And so we're interested in this sort of random mutagenesis approach because it can basically capture the whole genetic landscape that is driving a phenotype. And so we generate these large libraries of different mutants and are testing these mutant genomes in essence. And the first story I want to talk about, which also sort of got us into this whole area, is the question whether we can actually find new pathways that enhance protein homeostasis. As I said, we're interested in protein quality control, and whether this is sufficient for lifespan extension. And so a genetic screen, as we do it, uses EMS, the chemical mutagen, and then 2 generations removed, we can actually screen for phenotypes we're interested in. And in this case, we screen for resistance to a drug. This is a drug that perturbs protein homeostasis. It's called Tunicamycin, and it specifically acts in the ER and perturbs protein folding there, and is therefore highly toxic to worms. So when we take normal wild-type animals, they do not develop on Tunicamycin, but here we're using mutants that now, due to these mutations that I mentioned previously, randomly receive the capacity to actually become drug resistant and to develop on this toxic dose. And here we're basically made of mutants that are more stress resistant. And the question, of course, was whether this enhances now or enriches for mutants that are also long-lived. And this was indeed the case. After systematic lifespan analysis, we sequenced the whole genomes to actually identify the causal genes for this longevity. And I have to point out here that we're here using a proxy. So the thinking is that, you know, it is well known that stress resistance is linked to longevity. So can we actually make a stress resistant worm that is then long-lived? So it's a proxy for longevity. We did whole genome sequencing to identify causal mutations, and the pathway that we found and the enzyme that we were most interested in is called the hexosamine pathway, and the key enzyme of this pathway is fructose 6-phosphate amidotransferase, or GFAT for short. This was interesting. Yes, please.
Could I just ask a short question? This is
linked
to this mutations that you randomly introduced. So how do you know which mutations are related to the phenotype?
We don't.
You don't.
Not at the beginning. Now, this is of course the challenge in this. But this is, for example, this is a good example here because we have to sequence the genomes of multiple animals. And once we find sort of a convergence of the different mutants on one pathway or one gene, that is a very likely candidate to actually be causal. So I'm pointing this out specifically that we have this sort of shotgun approach, but then after sequencing, we have like maybe 50 or so point mutations in each genome. But if we then have, like, for example, 10 different mutants, and they all now have mutations in the same gene among their 50 independent ones, then we have a very good candidate to point to the idea that this gene might then be causal.
Okay, then introducing the mutation later on to kind of prove the theory.
Of course, yeah. No. This is basically just discovering a candidate, and so we use CRISPR-Cas9 then to actually reintroduce this mutation into a clean background to actually show causality. Okay. So these screens are just a basic starting point. Okay, so in this experiment, we identified, and this is exactly what was just the recent question, we identified independent mutations in this enzyme GFAT-1. So we had mutants that were stress resistant and long-lived and carried a mutation in GFAT-1, and we had 8 independent mutations in total that hit GFAT, and each occurred by introducing a single amino acid substitution, which was very interesting to us. Also, this was then the gene that had the most hits across the screen, so making it a strong candidate. Okay, so the hexosamine pathway is a metabolic pathway, which is then controlled here at this first step by GFAT-1, and it produces, using fructose 6-phosphate and a number of other high energy compounds, produces UDP N-acetylglucosamine down here, and UDP glycnac is actually utilized to synthesize glycosylation precursors and glycosaminoglycans. And this process is actually inhibited by tunicamycin, the drug that we used for the screen. What's also important to know is that UDP glycnac directly binds to GFAT-1 in an inhibitory feedback loop, basically being a very well-defined, well-behaved metabolic feedback where the product of the pathway actually inhibits the key enzyme. And so idea was early on that this pathway might actually be activated by the point mutations because it might counter the effect of tunicamycin. And this was indeed the case. We showed this in numerous ways, but one of them was to actually measure UDP glycnac, and we found that the mutants, actually we analyzed 3, it's true for all of them, actually had higher levels of UDP glycnac, suggesting that really we have a gain of function here, which is also nice because it shows that single point mutagenesis can actually give us not only a loss of function, which were used to in genetics, but rather we actually had a gain of function. And so in the end, these are some of the mutants we looked at. They are actually long-lived, and actually not only are they long-lived, but also they are protected against toxic proteins, where we can, for example, express an alpha-synuclein in these worms, and this actually improves their ability to move. The next question we asked was, whether these point mutations that we found in the worm might also be relevant in higher organisms. And I just want to go back and point out how highly conserved GFAT is. These are the amino acid sequences around the mutations, but also across the whole enzyme, it's very highly conserved. So we were wondering, does it have a role in protein homeostasis also in cells? So we introduced the same mutation that elevated function in the worm into the mouse, the locus in the mouse cells, into A cells, and this G451E substitution then also increased UDP GLICNAC levels in cells, as did the supplementation with the precursor of the pathway. And what's really particularly cool is that we could actually find a reduction in a disease-associated protein. So here we're using another aggregation-prone protein, ataxin-3, and ataxin-3 usually is aggregating in cells and is therefore present in this so-called formic acid soluble fraction. But when we actually point mutate just this one amino acid in GFAT-1, we see that the formic acid fraction of toxic insoluble protein is largely gone. So this shows that the control of protein homeostasis by the hexosamine pathway is something that's potentially conserved. Going forward, we tested whether this would also be true for survival in mice. And after a very long experiment, we actually found that both that the supplementation or the genetic activation, the supplementation with GLICNAC or the genetic activation of the pathway did not, in fact, enhance survival in mice. So in our experiment, for reasons that we still need to understand, lifespan was not affected. And this is potentially a very common and expected thing, because as you go higher in complexity, there are, of course, more things that can go wrong in an organism. And so it's a good example how these tests can just bring you very clear data. It's maybe not exactly what one had hoped. Nonetheless, we also looked at the behavior and particular learning ability in these animals and used the so-called Morris water maze experiments, which looks at spatial memory in mice. These mice have to swim in a large bucket of water, which they don't like, so they learn how to swim to a safe platform. And this learning can then be actually quantified. And what we found is that specifically males, after they were supplemented with GLICNAC in their drinking water, had a better memory performance, because they were able to be faster at the safe platform and actually visit this platform more often after having this diet. So despite the fact that they don't actually quite live longer, there seems to be a beneficial effect to brain function. And the weird thing that we're still really trying to understand and sort of begin to wrap our head around is the question why that is now specific to males and not to females. So this is sort of the direction that we took in terms of our question for the hexosamine pathway in vivo. But I want to go back to a much more higher resolution here and go back to the protein sequence. So we noticed that, as I mentioned before, individual amino acid substitutions can activate this enzyme. And we figured that having this information, which actually alters function in this information about amino acid substitutions that alter function in this protein, this might actually tell us something about how this protein works. So we were lucky and were the first ones to actually solve the crystal structure of human GFAT-1. Human GFAT-1 consists of two separable domains, a glutaminase and an isomerase domain. And what was well known and understood previously is that UDP GLICNAC actually is visible right here, directly binds to GFAT-1, as I had mentioned before. So this is an inhibitory interaction. But what's important to know is that UDP GLICNAC binding actually inhibits the activity of the glutaminase domain, and that active center is down here. So there's an intramolecular communication that has to occur to inhibit the wild-type protein function when UDP GLICNAC binds. And what our point mutations now reveal is really a lot about how this enzyme actually works, because it is some amino acids that are positioned here between these two domains, and this is the G451E mutation that I had just shown previously. in cells that now seems to interfere with this inhibitory feedback loop. So what we think is that the gain of function we have in GFAT is really loss of this feedback inhibition. And this feedback inhibition is actually not lost because this product is unable to bind. It can bind very well, we showed that. But this intermolecular communication, this communication of binding here, inhibition there, that is disrupted. So we learn a lot about how this enzyme in fact then works. This is of course not speculation only, we actually measured it. This is the wild type enzyme that is nicely inhibited by UDP-GLICNAC in a dose-dependent fashion. And the G451E, so just this one point mutation, is actually able to completely block this feedback inhibition. And so over here at these concentration areas, these concentrations, we actually have then a functional gain of function in this enzyme. So I'll just cycle back really briefly and say that wild type GFAT is inhibited by UDP-GLICNAC, and the point mutations that now randomly occurred from this entire genomic screen actually pointed us to this intramolecular conversation, I wanted to say communication within this enzyme that results in the wild type inhibition, and that inhibition is then lost. And we showed that because the inhibition is completely gone. Inhibition is a reduction in function in an in vitro assay, and the G451E, so just one amino acid substituted, basically makes the enzyme completely blind to this inhibitory function. And that's kind of cool, right? Because we get from a whole genome screen where we ask, can we find mutants that are stress resistant and then also long-lived, to a particular amino acid that results in this pretty large physiological change. And we also then wanted to leverage this knowledge to actually find small GFAT activators or inhibitors to actually modify this process with small molecules, and that's an ongoing collaboration with the Lead Discovery Center in Dortmund, which is a Max Planck sort of screening company. Okay, so I just want to go back and just one particular important detail in genetics that makes screens very interesting and is an important point here. And that is that if you do genetic screens normally, it's very difficult to find recessive genes, because when you do point mutagenesis the way we do, actually one typically loses the function of one gene, whereas the other copy of that gene is normally maintained and can then rescue that function. And thereby, in diploid systems, there's always sort of a copy of the gene there that can mask phenotype. And this is something that is actually circumvented in part in the worm and also in yeast, but also, and we developed this now, in any haploid cell. And so if you have a haploid cell, there's only one copy of each gene in the genome, and therefore then just hemizygous mutations such that occur only in one gene actually can lead to direct direct phenotype. So we have a 100% genotype to phenotype correlation here in these haploid cells. And so this is nice, and we then actually developed this, and we're lucky that haploid mouse embryonic stem cells had been recently derived. And these are really cells that shouldn't exist, right? Because haploid cells, it's only really egg and sperm that's allowed to be haploid in our biology. But actually it's possible to isolate haploid cells after a specific treatment from developing mouse embryos. And what one does here, and this is what this team in Josef Penninger's lab has done, is to basically FAC sort an inner cell mass of a developing embryo and simply FAC sort a 1N population. So normally cells go from a 2N stage, 2 copies of each chromosome, to the 4N stage, and then back to the 2N stage during cell cycle. So normally you have 2N and a mixture of 2N and 4N cells in a dish. But if the cells are haploid, they cycle between 1N and 2N. And so by simply FAC sorting to the 1N population, one can really then purify these haploid cells. And these haploid mouse embryonic stem cells are really quite amazing because we can culture them very easily. And each cell only carries one copy of each gene. That's their magic. So you can already sort of predict, I guess, what we did. we actually used again point mutagenesis here by in order to generate the same sort of library of mutated genomes depicted now here by these mutagenized cells. However, compared to the worm, we can do this mutagenesis here a lot deeper. So in worms, we can perhaps generate 200,000 mutagenized genomes, which is a lot of work and a very large screen. But here we can go up to 50, 60 million cells and mutagenized genomes in just a day, which is really quite remarkable when we now then do these amino acid resolution screens. Here we did again screens for toxic compounds and selected resistant clones that then can grow up in the presence of a selection, and then we can identify and pick these clones. And again, what we did is to do a screen for tunicamycin resistance. And what we found this time is actually another enzyme. So we were sort of expecting to find new mutations in GFAT-1 or GFAT-2, which is expressed. But in fact, what we found is a deacetylase that actually catalyzes another step of the hexosamine pathway. And this is sort of striking, but if you think about it, it makes total sense, because we get tunicamycin resistance in the worm by the gain of function of GFAT. And in these cells, we got tunicamycin resistance by loss of function of a reverse reaction. So this enzyme AMDHD2 is a deacetylase, and it mediates the reverse reaction from here up to here. And so it's the same net effect. Either you have a strengthened forward reaction or a weakened or strengthened reverse reaction, a strengthened forward reaction or a weakened reverse reaction in this AMDHD2 knockout. And in fact, we showed that we generated, and this also answers the previous question, we generated knockouts by CRISPR, and we can show that these are actually tunicamycin resistant and they have more UDP-GLICNAC. So this tells us something very interesting about these cells. But we also wanted to understand what AMDHD2 is. It's sort of an enzyme that we never had on our radar very much. And we solved the first crystal structure of AMDHD2. This is again the human AMDHD2. And we found again that many of the point mutations, now we have here multiple single amino acid substitutions from this screen that lead to the phenotype. They all sort of cluster in an active center and tell us a lot about, then again, how this enzyme works. And we also found something how these embryonic stem cells work. So in this case, now we're not using them in the haploid, but in the normal diploid state. And what we noticed is that these cells, if you look here in the Western blot for GFAT-1, express almost no GFAT-1. So this is the mouse embryonic stem cell line we use in the haploid state, and this is another mouse embryonic stem cell line, and these are some other cells. And the stem cells actually have no GFAT-1. So no surprise we didn't find GFAT-1 mutations, right? It's not really expressed. But what they express and the enzyme that they use to actually fuel the hexosamine pathway is GFAT-2. And GFAT-2 is then not expressed in these other cells, suggesting some sort of a switch, some sort of configuration that cells use in the hexosamine pathway that is specific to embryonic stem cells. And so we were wondering why that would make sense, and looked at the kinetics here of GFAT-1 versus GFAT-2 inhibition. So you're familiar with this now. GFAT-1 is nicely inhibited by UDP-GLICNAC, but GFAT-2 is actually much weaker inhibited by UDP-GLICNAC, really suggesting that feedback inhibition is really not a big choice that these cells have when they actually drive the hexosamine pathway through GFAT-2, and therefore the reverse reaction somehow becomes much more important. And then we have basically a forward reaction through GFAT-2 that's now not very much inhibited naturally, and then we get elevated UDP-GLICNAC by losing AMDHD2. I hope this makes sense, but the point I'm making here is really that using these screens, we get this really mechanistic insight into how these cells are wired, something we really hadn't expected before at all. And this is really the power of these forward genetic approaches. All right, I want to switch a little bit now, but stick with the theme of genetics and protein homeostasis. But if there are any questions up to this point, I'd be more than happy to take any.
Hey, Martin.
Hey, Maribel, good to see you.
Hi, good to see you. Yeah, I used to be a student in Martin's lab back in Germany, so that's how we know each other. So I was just wondering, basically, in any case where you supplement any basically metabolite from further down, do you basically then expect inhibition of something that is further up, or basically and then, as you said in the mouse, you basically did that, but then it's maybe too complex to see any phenotype for it.
Yeah. Well, I mean, it depends on it's a very good point, because you can uncouple these mechanisms by sort of feeding downstream to sort of replenish a pathway. So that's definitely a possibility. But these pathways are really complex, and obviously one thing that we find here is that they have this alternative configuration that really rewires the pathway just in the first two reactions. And so we think that just that actually has some specific and significant consequences. So this gets a little bit geeky, but you asked, right? So here's the idea. Look, what I think is actually happening is that in stem cells, since GFAT-2 can't really be inhibited, we have a strong entry into the hexosamine pathway that's always on, if you will. But then at the same time, AMDHD2 is also always on. So I think what's happening is that we have a cycle which actually is quite wasteful. And there's some reason why embryonic stem cells will have this, and something that we're actively working on. So this is a big ongoing question. And so this points to an important function just in this upper sort of regulatory part of the pathway, the first two reactions. Maybe they don't touch on UDP-GLICNAC so much, but in fact this pathway also consumes acetyl-CoA and glutamine. So they crosstalk to different organelles, different pathways. You can all imagine it's within the metabolic network. This is probably going to shift many balances around. So this is something that we're actively pursuing actually right now. I don't know if this is really an answer to your question, but it's definitely something that points to the specificity in a certain cell type, and how that now might actually be relevant to different cell types in an adult mouse is completely open.
No, that was really good. Thank you. I think it's like, it must have been like a sort of surprise when you did it in the haploid cells and realized that you can't achieve inhibition, basically.
Exactly. It also made us feel a little bit stupid, because only after doing this whole experiment, we actually looked by qPCR if GFAT-1 is expressed, and found that it's actually GFAT-2 that's expressed. So, sort of nicely fell into place after a while there. Yeah.
Thanks.
Right. Going forward, I want to go back to the worm. And again, I pointed out previously in our screen with Tunicamycin, we were using a proxy phenotype, right? We used Tunicamycin because we thought that finding resistance to Tunicamycin might actually enrich for mutants that are long-lived. But I found that sort of myopic, because it's sort of we find what we expect to find sort of situation. And so what we wanted to do when I actually started my lab is to actually do a screen that looks at longevity directly. So not any other lenses to go through, not any other selectors, but really ask, can we do this mutagenesis approach as I previously laid out, and can we find long-lived mutants as a first step? This is technically challenging, which is probably also why nobody had really done it in a large genomic scale. Some first experiments were published quite a long while ago, but we actually, thanks to having a whole genome sequencing available, were able to make this whole genomic approach. So we could really ask, what's the genetic landscape of aging, and can we find some longevity genes that are not known yet? And to make a long story short, we actually found not only one enzyme, like GFAT in this previous screen, but now we found the enzymes that sort of are functionally relevant and functionally combined in a pathway. And that pathway is again relevant for protein homeostasis, which was kind of cool to realize, right? Because we have an interest in protein homeostasis. And the pathway, when we actually look at longevity from a whole genome, unbiased approach, is again one that controls protein homeostasis. And this is the pathway that controls protein biosynthesis. This is a pathway that is controlled by the translation initiation factor EIF2. So translation initiation is really the rate limiting process that controls ribosome activity and protein biogenesis. And eukaryotic initiation factor 2 is actually activated by a guanine nucleotide exchange factor that's called EIF2B. And this then, under plentiful conditions, can lead to translation activation. So this is essentially the signal for ribosomes to go produce protein from reading mRNAs. Now, this pathway is, of course, extremely costly, right? Protein biosynthesis is the most costly thing a cell can do because it obviously needs huge biosynthetic machines that make huge products such as proteins, which cost a lot. And therefore, it's very tightly regulated. And one regulatory pathway is the so-called integrated stress response, which essentially senses different types of stress and integrates the stress information on the phosphorylation of one particular residue of EIF2. And this is now stress in worms, for example, that is sensed by GCN2, which is sensitive to depletion of amino acids, or PEC1. This is the PERK homologue of the worm, which actually reports on ER stress. So these are the inputs that worms that protein initiation of translation receives from stress in the worms. And so when the stress level is high, phosphorylation of EIF2 alpha is high. And this EIF2 alpha phosphorylation actually makes EIF2 an inhibitor to EIF2B. So now the EIF2B EIF2 goes from being a substrate of EIF2B to being an inhibitor. It's really remarkably structural, it's very well understood, and is a remarkably effective way of shutting down protein biosynthesis. Okay, so now I talked a lot about protein biosynthesis. Why is this relevant? This is relevant because in our screen, we actually found mutations in a subunit of EIF2B, in the gamma subunit, in GCN2, and in PERK. And this was really striking because these act in different ways, right? The EIF2B sort of promotes protein biosynthesis, whereas the kinases actually counter protein biosynthesis because they activate the ISR. So we were really scratching our heads, but it was really nice to go from this genomic approach to just look which process is actually affected most. And I'm not kidding you, this is really the process that sort of went to the top because we had these independent mutations across these related genes. Okay, if you're working in worms and you find mutants that make the worm long-lived, that in any sense smell like they have to do with protein biosynthesis, the first hypothesis is always, well, we have a reduction of protein synthesis, because it's well known that reducing protein biosynthesis extends lifespan. This is very well conserved and is certainly the case in worms. So we looked at protein biosynthesis, and this is a method that's called ribosome profiling. It basically uses a very elegant gradient centrifugation method that separates ribosomes sitting on their respective mRNAs. And you can basically separate monosomes from disomes, from trisomes, and so on, and basically get this very typical absorbance peak, where you basically have the two ribosomal subunits by themselves on the mRNA. There's a monosome peak here, and then you have basically the peaks that correspond to sort of 2, 3, 4, 5, 6 ribosomes on one mRNA. And the idea is that the mRNAs that have many ribosomes associated with them are actually most actively translated, simply because they're more ribosome associated. So now the mutant that I'm showing here, PPP1, that is just worm speak for the gamma subunit of EIF2B. And we have 2 independent mutations in this particular gene. And there was absolutely no difference in protein biosynthesis in these worms. This was very important to show because you would expect that actually you lose protein biosynthesis, thereby worms become long-lived. These worms had no problem with their protein biosynthesis in looking at their bulk. So no changes here. So it must have been something more complex and perhaps more interesting. And to understand what's going on, we in fact asked, are the proteins that are actually produced, are they in any way different? So what we did is we actually specifically isolated the mRNAs that are associated with these higher order polysomes and simply asked, are the and then simply sequence those mRNAs. So you basically get a measure for translational efficiency by doing this. So we basically have a whole transcriptome, and then we sort of use that to normalize the signals we get by sequencing only these polysome-associated mRNAs. We found that a few of them were actually de-enriched, some were enriched, and then some particular ones we actually then submitted to additional screens, and then found one particular kinase whose translation was significantly elevated, while the mRNA abundance was not changed in our mutants. And we asked then if this kinase that we called KIN35 is relevant for the longevity. This is the longevity of these worms. PPP1, again, is this gamma subunit here of EIF2B. And the solid green and blue lines show the longevity of these mutants. Very nice, and also helpful to have these 2 different alleles. But then when we do RNAi against KIN35, lifespan basically falls back to the wild type. But wild type itself is actually not shortened by KIN35 RNAi, which means that it's somewhat specific to our phenotype. So obviously, KIN35 is selectively translated in this condition and somehow makes these worms more long-lived. And we're still trying to understand what KIN35 is actually doing. Okay, so to sum up, what we found here is that these mutants all sort of modulate the integrated stress response, and that is that they inhibit it. So we showed that the kinases actually have reduced function, so we have less stress input into the pathway in these mutants, and this EF2B mutant is actually genetically dominant, suggesting that it is stronger in promoting this translation initiation side of the pathway of the integrated stress response, suggesting that really what we're doing is we're tuning the whole pathway to inhibit the integrated stress response to benefit this side over here. And we can do this by losing function here or adding function here. So it's the same net effect that these mutations have. We then went on to ask if these, then I have to mention this. We are very excited about these findings because there's a very interesting compound out there that is actually an activator of EIF2B. And this compound is called ISRIP, Integrated Stress Response Inhibitor. And this compound actually, and this is work that's been shown by Peter Walter and many other labs, actually is able to counter the pathology related to numerous neurological conditions and neurodegeneration as well. So inhibiting the integrated stress response seems to be something that's very beneficial in the face of chronic stress. And so we tried actually to ask if this would have an effect on survival. This experiment is still ongoing, but I can tell you that these mice actually, again, using a Morris water maze, when we feed them with ISRIP, they're able to learn, but they're also, if we look at this memory retention after 12 days, they're also a little bit better at actually remembering this safe spot when we remove the safe platform in this memory assay, suggesting that their memory function is somewhat enhanced, and that would be very nice if that was to be also true for the lifespan, and where the lifespan then would be extended in these animals, and that's still ongoing in mice right now. All right, so I think here are some future perspectives, but I don't want to bore you too much with this because I think I've sort of made these points fairly well. And before I leave, I just want to introduce a quick system, just really briefly, that we've now developed, and that is to do ribosome tagging. And you see that we've developed a very keen interest in protein biosynthesis and in mRNA translation. And what we've done so far, and specifically in this PPP1 project, is to simply just take all ribosomes from the worm. But of course, the worm has many different tissues, and therefore, there's really a need to do polysome to look at translation in a more tissue-specific manner. And we can now do this by actually adding a tag to RPL22, to the large ribosomal subunit, and expressing these tags now in specific tissues of the worm. And you can see this here. So we have these very nice transgenic worms. This was developed by Amy Prukic in my lab together with Laura Vesta and in collaboration with the Mayer Lab in Harvard. And the very cool thing, and I just want to show this here now as a quick teaser, is that when we isolate these RNAs now from these translated RNAs from these worms, we can actually see very big differences. So it's very nice to see that when we simply express when we simply look at the total mRNA, the different worms, of course, don't differ because they only differ in the expression of this tagged RPL22. And instead, when we express the tagged ribosomes in all different tissues and then sequence the associated mRNAs, they differ quite a lot in this principal component analysis, which really makes us think that we have a very specific system here, and we can now ask very interesting questions when it comes to control of mRNA translation with age, for example, or with different treatments in different tissues. All right, I want to come to an end now. I'll be more than happy to take some questions, but first I want to point out the wonderful team that I have. This is the group that I've had the pleasure to work with over the last few years, and this is actually a hike we did right after we could again have a retreat after COVID. So this was really nice, and they've really powered through this really difficult time. And many of them are now going to relocate to me to the UK. This is my funding. These are my collaborators, and yeah, thank you so much for your attention.
Hi Martin, thank you so much for the talk. I really enjoyed it. I really enjoyed the deep delve into the mechanistic causings of aging and longevity. And yeah, thank you so much. And yeah, I really liked your experimental approaches as well. So yeah, again, if anyone has any questions, feel free to put them in the chat or just unmute yourself, like whatever you find easier.
Would you mind if I start with a layman question, actually, or maybe an easier question for you? I saw that in the x-axis on one of your plots, you had the maximum age that is currently, or that was shown there of this C. elegans worm was 40 days.
Yes.
Could you tell me, like, what is the maximum achieved so far?
Oh, in worms?
Yes.
There's some crazy long-lived worms out there, so this is something that we haven't really achieved yet. But if you combine different parameters, so it's known that males, for example, live longer than the hermaphrodites, and then by using specific mutations in the insulin signaling pathway, these worms become really, really long-lived. So up to 3 months is actually possible. So then there are also different stages where during development, these worms can actually basically pause their development or develop through alternative larval stages that actually don't age. So they can actually remain there for a very long time and then have a normal adult lifespan. So lifespan is very plastic in these animals.
Interesting, thanks.
Hey, just jumping on that question as well. Amazing talk. I was also curious, what do you think is the most important bottleneck limiting C. elegans lifespan?
Yeah, I mean, that's an interesting point. What's the bottleneck? I mean, it's the question is sort of whether you have one particular pathway that's sort of most limiting. That's, I guess, where you're coming from, or if the whole, if you know, through hormonal regulation and coordination between tissues, there's sort of a decline that affects the whole worm, and there's evidence to support either. In a way, the screens that we do sort of go for that, right? Because because we go doing this in this unbiased fashion, we do have ways to modulate just the process that is sort of the first one to extend lifespan, right? But on the other hand, I wouldn't say that now, you know, changing, for example, the tone of the ISR does this, because that then crosstalks to many other things and potentially in other tissues too. So that is somewhat coordinated. So there, I don't think there's one thing. Worms, if you just watch them on their plates, they really do look old after a while. So it's all their tissues that sort of become less functional and less organized. So it's hard to pin it down to one thing.
Okay, thank you. It seems, do you think that you're mostly finding mutants that are alleviating ER stress rather than extending longevity?
So in the Tunicamycin resistance screen, it's kind of an interesting thing that we realized after a while, actually, because the mechanism by which worms become resistant to tunicamycin is a different one than the one that actually makes them long-lived. So both are linked to elevated UDP-GLICNAC, that we know, but basically UDP-GLICNAC just simply outcompetes the function of tunicamycin. So it's a very sort of physical competition at the level of a specific target enzyme. Now, the fact that these UDP-GLICNAC elevated worms are actually long-lived was sort of really a beneficial side effect, which is why we then picked it up, right? And so actually UDP-GLICNAC actually triggers, or this elevation of UDP-GLICNAC triggers a number of processes such as autophagy, and also the proteasome is more active. So a number of degrading protein degrading processes are actually enhanced in these mutants, and that's how they're long-lived. So in fact, to come back to your question, we're not seeing a direct effect on ER physiology in these worms, but these worms are now, when you take them just on normal growth media, they just have more UDP-GLICNAC, and what happens there is then downstream to that, and that includes but isn't limited to the ER.
I have a question. So apart from like nematode worms, what other organisms would you be looking at to carry out like similar like mutagenesis screens?
Oh, I like that. In a way, the nematode is super interesting, and people probably are right in saying it's not a model organism, but it's an organism, right? Because you can find really interesting biology there, and also the basic biology that we're looking at is one that's shared across all organisms in a way. So the power of the worm is really that the genome is so well understood, and that sequencing is now available. So I think these are the ingredients for these screens. So we can keep them sort of at really like homogeneous at the level where the genome is very homogeneous, because they're all basically clones of one another, because they're hermaphrodites. And then we can do chemical mutagenesis and sequence genomes. And the other thing is that, of course, in these screens, you want a phenotype that you can easily see, like tunicamycin resistance, right? It's like a yes/no answer. And so any organism that is capable of bringing these attributes with it are useful. Maybe another point is time, because you don't want to wait forever for these animals to show phenotypes. But I mean, they've been amazing and very famous screens for development in flies, right? Where Hox genes were essentially found. There's a very similar approach, but just looking at developmental patterns in the fly. And even in mice, there's large mutagenesis approaches ongoing, where then people have very many mouse lines that have small differences in their genome, and one can then look at phenotypes. Of course, we're not patient enough for that, and that's why we're working with the worm. But that's sort of things that one would consider. So before taking in a new organism, if that's what you're planning to do, you really need a good genome sequence to do that, and some good basic understanding of the organism.
Yeah, so I guess in that sense, like Drosophila would be quite good for that.
Sure.
Yeah. What
Yeah.
about like planarian worms? Like, have you done like the same sort of like mutagenesis screens, or is like the
No.
mapping not too good for that?
No, I don't know. I don't know if I'm not very familiar with that field, but I would think that people have used this opportunity that these high resolution screens afford, particularly because you don't need to have like something like CRISPR developed, right? Because with CRISPR you can do similar, but let's say overlapping things. But I don't know about point mutagenesis there. It would be very interesting.
Yeah, it would be, but I really wouldn't know like how far like the mapping of the planarian genome has gone. But yeah,
I don't know.
Yeah, but I guess I think the main point is if you take those organisms with like a really well mapped genome, then you can like identify like the mutations much better, and therefore
Absolutely
find the mechanisms. And then you can find, like those robust mechanisms from that. So yeah, I really like the sort of mechanistic approach in this. So yeah, I really, again, just enjoyed your talk. So thank you so much for
absolutely.
Yeah, hopefully maybe you'll be back soon. Yeah, I guess.
Yeah, I would love to do this in person. Now, in the future, I won't be too far away from everyone in Oxford.
So yeah, definitely we'll invite you back.
That would be great.
Yeah. If there's any last minute questions.
I have a quick question.
Okay.
Congratulations on a very interesting presentation.
Thank you very much. I really appreciate it.
Do you have any line of work that you want to open up once you move to Cambridge, or you want to continue with this?
Both. So I think the fact that at Altos we have just really fantastic colleagues and a whole new team of researchers, I think there will be huge opportunities in that. And my lab has really developed towards this control of translation initiation, as you saw from this presentation. So we're going to continue on on the one hand, this sort of metabolic control of protein homeostasis, but then we'll develop more tools to really look into the control of translation initiation, of which some I showed here. And I think there's really a lot to do and to understand, particularly in the worm, which is just a fantastic organism for that, because the somatic cells of the worm, they're post-mitotic, right? So we can really study this in a system where cells don't constantly divide, which is the case, of course, in tissue culture. So we're trying to use the paradigm there for those purposes.
Excellent. Thank you.
Okay, just thank you again. And yeah, hopefully, Martin, you'll be back in Trinity term. Let's organize something.
That would be wonderful.
Yeah,
absolutely.
We'd love to have you. So yeah, thank you very much. And everyone have a great evening. Enjoy the rest of your weeks. Thank you so much.
Bye-bye.